Welcome everyone. This is our Global Health Innovator Talk series from PathCheck. We are focused on crowdsourced computational health solution. If you want to know what we are doing, you can actually visit on pathcheck.org. You can look into our analytics solutions, which is available on dice.pathcheck.org. If you want to know that, if you want to connect with us, want to work on volunteering solutions, please do reach out to us on tiny.cc/pathcheck Slack. And if you want to connect with us on research talks, please do reach us out at tiny.cc/pathcheck PC research talks. This research talk is organized by the PathCheck community, and a few of the leaders who are mentioned over here are Ramesh, Rohan, Shanice, Graham, and Nina. They are primarily the leaders in PathCheck research talk organizers, and they also help us to organize these research seminars. So what are these talk series? Research meetups are basically to create an impact on digital AI, and we are looking into digital health, and we want to understand that how AI software technology can create an impact on digital health. We also look forward to driving collaboration on AI for health with cross-disciplinary efforts for computer science, medicine, bioinformatics, and more. With that, we would like to introduce you to Dr. Fergus Imrie. Dr. Fergus Imrie is a postdoc at the EC department in UCLA. He is motivated by the promise of transforming healthcare and improving medical knowledge through the use of machine learning in combination with clinical experts. He's also interested in self-supervised learning and methods for understanding clinical decision-making. Prior to joining the lab, Dr. Fergus have completed his PhD at the University of Oxford in the Department of Statistics, where he was developing deep learning approaches for drug discovery. He's also interested, and his research is currently being used by a number of pharmaceutical companies on active drug discovery projects to develop new therapeutics. So without further ado, we would like to introduce you to Dr. Fergus Imrie. So Dr. Fergus, if you can start sharing your screen, and we can initiate with our research seminar series.
Great, thank you. Thanks for that introduction. Let me just share now. Great. Does everything look okay?
Absolutely fine.
Fantastic. So thanks again for the introduction, and thank you also for the invitation to speak here today. I'm absolutely thrilled to be here. So really do thank you for that. So today I want to speak about self-supervised learning for omics. And while the application focus of what I will discuss today is about omics data, the methods I'll discuss aren't specifically limited to just this data type and can be applied to other clinical data types as well. So I want to begin by highlighting perhaps the key driver behind what I'm going to speak about, and what will likely be one of the most important factors that I believe anyway will change medicine in the future. And that is firstly the ability to sequence the human genome, but perhaps of much more practical importance, the cost of sequencing the genome. So while the estimated cost to sequence a human genome around 15 years ago, at the end of the Human Genome Project around 2006, was over $10 million just for one single genome, less than 10 years later in 2015, it had already dropped by over 100-fold down to below $1,500. And now it's only a few $100s to sequence an entire genome, and even less than that if you only want a specific genetic test. And as a result, we've seen a great increase in the number of genomes being sequenced, both through government-funded projects, such as the 100,000 Genomes Project of Genomics England, but also through retail channels and through genetic testing through private companies such as Ancestry.com or 23andMe, amongst others. And I really want to highlight that this relative inexpense of sequencing allows for the routine collection of this type of data in all walks of life. And I think the one that I'm most excited about is in healthcare settings in order to improve healthcare for patients. But the real question is, how can we use such data to improve our clinical care of patients? And so there's a few things I'm going to talk about today. And the first is how we can use like omics and clinical data to develop polygenic risk scores that will enable us to deliver personalized medicine. I will also discuss how self-supervised learning can be used to identify biological associations to further our understanding of biology and help us to develop new therapeutics. So let us start with polygenic risk scoring. So the goal of a polygenic risk score is to predict the risk of an individual developing a disease based on their genomics. And the focus of polygenic risk scores is for complex or polygenic diseases that occur as a result of many genomic variations. And obviously these genomic variants don't guarantee you will or won't get a disease. There's also the fact that these are often paired with environmental influences. So for a long time, only linear models had performed well on genomics data for crafting polygenic risk scoring. But why was this the case? And our lab realized that only standard machine learning methods were being applied and compared against linear models. And also that the data really wasn't being used effectively to allow machine learning to tackle such high dimensional data as is needed for such a problem. So now let us understand why these previous approaches were failing and what we might be able to do about it. So we have high dimensional data, such as genome sequence as part of the 100,000 Genomes Project, where in the UK, around 85,000 NHS patients with a rare disease were sequenced. So at first glance, that might seem like quite a large number of samples, quite a lot of data. But for a particular disease, for example, a particular type of cancer, only really a small number of patients exist, and thus we actually don't have the amount of data we might think we have. And so our supervised learning problem, instead of being able to derive a risk score from a large labeled data set, where we should be able to develop a pretty decent model, we in fact only have access to a small labeled data set, which decreases our ability considerably to learn a performant model. So how can we overcome this? In many cases, we actually also have access to a large quantity of unlabeled data, perhaps from patients with other diseases. And this is where self-supervised learning can come into play. In order to conduct self-supervised learning, it adopts a two-stage training procedure, where first we use all of that unlabeled data to pre-train a model, and then we use this small quantity of labeled data that we do have in order to transfer the knowledge gained from pre-training on the unlabeled data, and we use the labeled data to fine-tune and update our model for the specific outcome or disease of interest. And I'll go a little bit more into detail later about the exact methods that we're going to use to do this. And so maybe as I presented it, maybe this sounds relatively simple, relatively easy. But the reality is that while self-supervised approaches have had a lot of success in other domains, such as computer vision for image recognition, or in natural language processing, in genomics or even medicine more generally, we're primarily concerned with a different form of data, and that is of tabular data. And I really want to emphasize that compared to these other modalities, tabular data is really unique and fundamentally different from those types of data. And as a result, the methods that have been developed for such data aren't readily applicable in this scenario, since they really do rely heavily on the inherent spatial or semantic structure of images or languages, respectively. To briefly explain what I mean by this inherent structure, I want you just to consider the image of the 2 on the right-hand side of the slide. And so the pixels that comprise the image are much more related to their neighboring pixels. You can see we've got these large regions of black, and then one of the white pixels that makes up the 2 is much more likely a pixels next to it is going to be white than a randomly selected pixel across the whole image. And this structure is used and exploited by self-supervised learning algorithms for images. However, in contrast, tabular data has no explicit structure. Just because 2 genes, let's say, happen to be next to each other in a list, doesn't mean they're related in any way. And as such, any structure in your data must be learned. So to address this, our lab developed a method called VIME that was presented at Europe's 2020 that introduces novel self and semi-supervised learning tasks for tabular data, including genomics data. So to explain how VIME learns from unlabeled data, consider the left-hand side of the figure first. So we first take an unlabeled data point, and then we corrupt this sample via a masking process that takes some of the entries at random and replaces them with other values sampled from the marginal distribution for that feature to get a new feature vector where, as I said, some of the entries have been corrupted to new values. We then train an encoder network to predict which values were corrupted by the masking process, known as mask vector estimation, and what the original values of those features were, feature vector estimation. So the goal of doing this process is that in order to achieve these tasks, the encoder must learn how the features are related to each other, and thus learns the inherent structure of the data. I note that then you can use the same masking procedure as a data augmentation technique for semi-supervised learning to increase the effective quantity of the labeled data after the self-supervision has taken place. So as an illustrative application, we applied VIME to genome-wide polygenic scoring. And we took a large genomic data set from the UK Biobank, and specifically we have around 700 SNPs after the standard p-value filtering process. And the goal was to predict 6 different blood cell traits. We treated these different traits as independent prediction tasks, and the SNPs are different for different tasks. To test the effectiveness of self and semi-supervised learning in the small data setting, the number of labeled samples we varied between 1,000 and 10,000, and the remainder of the data was used as unlabeled data, which was over 300,000 samples. And as you can see from the slide, VIME outperformed all of the benchmark methods tested, including state-of-the-art linear models for polygenic risk scores, ElasticNet. And this really shows the power and the possible potential for machine learning and deep learning, even in these high-dimensional settings. And we believe this is a very intriguing first step in developing deep learning models for polygenic risk scoring. In the remainder of this talk, I want to go beyond standard prediction tasks and move towards discovery and identification of associations. In a standard prediction task, we have features, which in the case of omics or genomics is normally quite a large number of features, and we wish to predict an outcome of interest. And so while, even in the previous slides, I've shown that standard prediction can achieve high accuracy, high performance, it doesn't inherently provide any additional scientific insight. It doesn't tell us why such a prediction was made, which features resulted in the outcome of interest, such as an observed phenotype, and what really drove the biological process. So what happens if we instead could remove the majority of features and make those same predictions based only on a small number of highly relevant features? This is now the problem of feature selection. And naturally, if we're able to achieve this, this gives us a much greater understanding of the drivers of the outcome of interest, and might also have other benefits, such as reduced cost if we only need to measure a small number of features, and also improved generalizability due to the fact that you're ignoring the majority of the features, and so you're less likely to overfit to such nuisance features. However, there are 2 significant challenges to performing feature selection in such high-dimensional data such as omics. And the first is that we've already encountered, which is that of limited labeled data. And without sufficient data, feature selection is likely to pick up on spurious relationships between features and the label. However, they're often unlabeled samples, allowing us to utilize self-supervision, similarly to what we saw with VIME. The second is intra-feature correlation, which can confound feature selection from choosing the variable that's responsible for the outcome. For example, let's consider the causal graph I've shown in the middle of the slide. And so, for example, let's say we've got a gene X1 that, when highly expressed, causes the phenotype Y to be observed in the patient. But high expression of gene X1 also causes gene X2 to be highly expressed. The feature we want to identify is clearly gene X1 in this example, since that is what is driving the clinical phenotype. But the presence of feature X2, which is highly expressed when gene X1 is highly expressed, makes this process very challenging. To address these two challenges, we've proposed a new framework for feature selection that we refer to as self-supervision enhanced feature selection, or SEFS for short, which will be presented at ICLR 2020. The overall aim of our model is to select the most important features, which is the job of the subset generator shown on the slide in purple. However, in order to learn how to select this subset, we will employ other network components and adopt a two-stage training procedure. In the first phase, in the first phase, we look to fix the subset, the selection probabilities of the subset generator and train an encoder using a similar objective to the approach of VIME that I described previously. And in the second phase, we fix this encoder network. And now we learn jointly both to generate these subsets and to select the features, as well as train a predictor network to predict the outcome of interest on the small amount of labeled data, all while minimizing the number of selected features. Perhaps the key component of our method is how these feature subsets are generated. And to tackle the problem of intra-feature correlations confounding the feature selection problem, we explicitly model the potential correlations between features via the use of a multivariate masking or selection process. Instead of selecting features independently at random, which is what previous approaches in feature selection have assumed, we select features using a multivariate Bernoulli distribution that selects highly correlated features together. In the self-supervision phase, this prevents the model from trivially predicting these features from the other features, while in the feature selection phase, it forces the model to select between the features which is more informative for the outcome of interest, so that you only get the most important features being selected. To demonstrate our approach, I first want to begin with a toy example on some synthetic data. So let's consider a data set where we have one relevant feature and 9 irrelevant features, but that are highly correlated with this first feature. On the left-hand side, using our proposed correlated gating procedure, we can see that the model quickly learns to identify the relevant feature and also not to select any of the irrelevant ones. In contrast, on the right-hand side, while using an independent selection procedure, the model learns to select the relevant feature. It also incorrectly selects several of the irrelevant features with high probability, generating the benefit of such an approach to reducing the inclusion of correlated, but ultimately less important features. So after this, we then took SEFS and applied it to several real-world omics datasets, such as proteomic observations from the CCLE dataset, where the target label is to predict the drug sensitivity to different drugs of various cancer cell lines. The dataset contained 899 cancer cell lines, of which only 458 are labeled. So this is really a very small sample size. And to benefit from self-supervised learning, we integrated the RPPA measurements from over 7,000 samples from the TCGA dataset. And so even though the measurements from these two datasets are collected in different conditions, our hope was that we'd be able to learn some underlying structure from this related data that could help us with this prediction task and to identify relevant features. So since there are no ground truth relevant features, really the best way to identify whether or not the features you've selected are of interest, at least in the short term, without extra experimental validation, is to see whether or not these features are predictive of the outcome of interest. And so in the figure on the left, we ranked the average prediction performance using the selected features for both our method and a large number of benchmarks across 10 plus drugs. And as can be seen in the figure, SEFS almost always was the highest ranked model and had the best predictive performance using the selected features selected by this model, outperforming many state-of-the-art feature selection methods. Obviously, prediction performance alone isn't enough, and one of our goals was to improve understanding. So the next thing we sought to do was to validate the discovered features in the scientific literature. And here I'm showing the features discovered by Cephs for 2 of the drugs, panabinostat, an HDAC inhibitor, and irinotecan, a chemotherapy agent. And we were able to validate almost all of the discovered features for both drugs. And in addition, many of these proteins were only identified with the version of our model that used self-supervision, demonstrating the importance of the self-supervision phase. We also applied Cephs to a transcriptomics dataset of peripheral blood mononuclear cells with the task of differentiating naive from mature T cells. And as you can see, Cephs significantly outperformed all of the baseline methods again, and similarly to the previous experiment, we're able to validate almost all of the discovered features in the literature, which was highly encouraging. So with that, I'd like to conclude that I believe omics is here to stay in medicine. And while it's already playing an important role in many diseases, I really do think we're only at the beginning, and so much more can and will be done. And one driving force, I think, for this will be self-supervision, playing a key role in tackling such high-dimensional, low sample size data. And with that, I'd just like to say thank you very much for your time. And if you'd like to know more, please feel free to either check out our papers, but also ask any questions that you may have now. And thank you.
Thank you, Fergus.
Yes, thank you, Dr. Fergus. Hi, I'm Graham Dodge. I'm the PathCheck Foundation president. I have to admit, I only was able to join about halfway through your presentation, but if I understood what you were talking about correctly, does this relate as well to sort of being able to provide prognoses using sort of a digital twin approach to medical efficacy in certain, as it plays out in certain genomics?
I think that so the digital twin you're referring to, so that's not something we've explicitly looked at or done in this context. But I see no reason why a lot of the techniques here couldn't also apply in those type of settings or be used in clinical trial data where you also have normally very little data and very, very cost driven in order to collect more samples there, where now that we have much more background data on just individuals, we might be able to include more genomic data there in order to better stratify different patient subgroups and maybe identify subgroups of patients for which drugs work particularly well or less well.
Yeah, I think that, to your point, more data obviously being put into these types of simulations, obviously the more effective and more accurate they will be. Fascinating stuff. Chris, do we have any other questions right now from the attendees?
Yeah, like I have asked the attendees to put their questions through, but just like Dr. Fergus, that was a very interesting, it was very interesting to see that how feature selection is done in the medical field and the healthcare ecosystem. So when I have a question that when you were working on SEFs that you're calling it, you looked into obviously the data set that you had, right? And you looked into the labeled data set and unlabeled data set. So for a real-time problem, like, if we want to use this similar methodology on imaging, right? So what will be the feature selection methodology when we look into these kind of feature selection, especially to identify or, early diagnosis of, let's say, cancer through imaging? So at that point of time, what do you think that the feature selection process could be? Or could we apply supervision enhanced feature selection on that?
No, thanks. That's a really, really interesting question. I think so. The goal of what we were doing was almost to stay away from imaging. And I want to almost highlight that imaging is quite a different problem. For example, if I take an image and the patient was moved almost like 1 pixel to the right, all of the individual pixels are now obviously shifted across, and so don't have that same meaning. There is no, I suppose, meaning to a particular pixel. Whereas in the case of tabular data, each feature has a specific meaning. Like, this is the measurement, this is whether or not this gene has a variant, this is the expression of this particular protein. And I think in that context, we can use feature selection to drive scientific discovery or identify new targets or do this, because when a feature is important, you know that, like, it tells you something about, there is like an underlying, I guess, real world thing to look back to. Exactly. That in the case of an image, what does a feature even mean in the case of an image? And I think there is progress that can be done there, but it's quite a different problem, because there, it's almost like you want to identify, like, one way feature selection could be used is in that setting is to say, okay, no, it's this part of the image that is kind of showing you've got cancer, and the rest looks normal, but this is the part of it. And I think that's a very different question in my mind, at least. I think self-supervised approaches will definitely be used in that context, where for example, you don't have very many examples which are labeled, but you have a lot of, say, non-cancerous cells, or you have a lot of potentially even more unrelated images. And I think there's a lot of promise there. But I think feature selection in that context is a very different problem with different aims and goals and outcomes.
That's brilliant. And what is the future of WIME that you have been working on this? So how do you see that the future of WIME will be in adaptation, like if you are adopting WIME, or if you want to adopt CEFs? So what is the future of it? Are you open for collaboration? Can people work? Is it open sourced?
Yeah, no, great question. So both methods are open source, sorry, I'll correct that. WIME is already open source. CEFs will be in the next month or so, in time for the conference that we presented at. And so that work is still kind of happening. We're definitely keen for future collaborations, and I think the future for both works. As you've seen, like, while we've used real world data in both cases, none of what I presented is kind of a medical study or has necessarily added to the medical literature as yet. And I think that's really where we want to take some of this next. Our lab is a machine learning, artificial intelligence lab primarily, but we do, and I guess the first step of that is developing new methods, but ultimately you want to see those methods through, and really they're only useful if they eventually impact medicine. And I think we've got some ideas, but definitely would welcome any interested potential collaborators to get in touch and discuss any topics they think might be impacted by these methods.
Absolutely great.
Dr. Fergus, we have a question here from Praneeth. Does your work take causal issues such as confounding into consideration?
Hi, that's a great question. We don't at the moment directly take these into consideration. No. There is a large body of work that does look much more closely at the effect of confounding, in particular for treatment effect estimation. But this wasn't something we looked to incorporate in either of these pieces of work.
So to follow on with that, so you don't necessarily require causal explanations when creating new compounds?
So I suppose we're not at the point of trying to create new compounds. I guess I'd view the feature selection work in particular as, let's call it a hypothesis generator for potential ideas. You've got 20,000 plus functional genes in the human genome. There's millions and millions of SNPs if you look across the whole genome. How can we have any hope of kind of identifying what is driving a disease? I think traditional approaches to this from the genomics world has used things like GWAS and statistical testing in order to identify correlations, not causation. So there's no causal effect in the current statistical tests being used for this type of data. but the type of associations such methods can pick up are limited to linear interactions and often very simple. This one gene has a statistically significant effect, like for a whole population. Whereas I think what we're looking to move to, and the promise of deep learning and maybe some of these other feature selection, is to be able to identify more complex interactions and to maybe change these statistical tests, which themselves aren't causal, into more with more powerful methods. So I guess the answer is almost we're replacing non-causality with non-causality. I think then more can be done, and further validation of some of these features. You do want to have a better causal understanding and identify, for example, where in pathways things sit. Does the expression of this does it feed to the, like, are you almost upstream or downstream in a process? But I think I can view that as almost like a downstream check versus at this point.
And we have another follow-up question about, can it be naturally extended to conditional correlation, as in conditional dependence?
I think that's a very interesting question. Something I'd want to think a bit more about, but quite possibly.
Well, I know that we are at our time now. Unless there's other questions, I will thank Dr. Fergus and the team here at PathCheck for putting this all together. Another great addition to our series. Thank you, Dr. Fergus. Thank you, Bethany Lamonaco and Krish for all your work and putting this together. And please follow us on all of our social medias for PathCheck Foundation to keep up with the work that Dr. Fergus is doing and others who are contributing to our Global Health Innovators series. Thank you all very much.
Thank you, Dr.
Thank you all.
Fergus.
Thank you, Dr. Fergus. Thank you, everyone.